Okay, so let's start with the introduction to DNA sequencing. Now, what is DNA sequencing? So DNA sequencing is the process of reading the nucleotides that are present in DNA. So basically, this is determining the precise order of nucleotides within a DNA molecule. So this is a very basic definition of what is it. And now there is a term that is DNA-seq that you might have heard of. And this generally refers today to any next-generation sequencing method or technology that is used to determine the order of the 4 bases, so A, T, C, and G, in a strand of DNA. So in fact, there are two main types of DNA sequencing technologies that are used today. One is the Sanger sequencing, and the other one is the next-generation sequencing, or NGS. Each of these technologies has utilities in today's genetic analysis environment, and we will be seeing the differences between the two technologies in the upcoming parts of this session. Now, the whole story began in 1953 when Watson and Crick discovered the DNA structure, but then it took almost 20 years to witness the beginning of the human genome sequencing with the first complete gene and complete genome of a bacteriophage. And then the real breakthrough in DNA sequencing was achieved in 1977 by the publication that same year of two novel sequencing methods, one from Maxam and Gilbert that used radioactivity, and this is what rendered it less popular than the second one, which is the Sanger sequencing that is still widely used today. So in 1987, Applied Biosystem marketed the first fully automated sequencing machine. And in 1995, it was the real start, I would say, of the genomic era when the first bacterial genome was sequenced. It was the genome of Haemophilus influenzae. And the sequencing was done using the conventional Sanger sequencing. So that procedure took about 13 months. And it was considered at that time a major scientific breakthrough, but the problem is that it was associated to high costs. So there was a need, in fact, to develop novel and more powerful technologies. And the first NGS technologies was published in 1996 and is called pyrosequencing, but the real acceleration in the efforts came after the sequencing of the human genome in 2001, which we will be talking about. And you have also here an overview of the NGS technologies that were developed more recently and that we will see later on. So what was this Human Genome Project? So this was a 13 years international effort to sequence the 3 billion letters of the human DNA. It is, in fact, difficult to estimate the costs that were associated to that project for the release of the draft sequence, but it was certainly in the many tens of millions of dollars. You have here an approximation of $300 million project. A draft was produced in year 2000, and the Human Genome Project then proceeded to refine that draft genome in order to produce what they called a finished human genome. And it was an effort led by a group of publicly funded researchers in an international human genome sequencing consortium called IHGSC. So, in fact, before the public consortium published the first phase of the Human Genome Project, there was a private company that was called Celera Genomics, led by Craig Venter, that also announced in 1998 its will to sequence the human genome. So, what Craig Venter wanted is to sequence the human genome as quickly as possible in order to accelerate the search for disease cures. And I would say, unsurprisingly, this initiated a competition between both the public and the private sectors. And the real problem came from the fact that Craig Venter aimed to only make the information available to the paying customers, planning, for example, to patent gene sequences before their release. So basically, you would have had to pay in order to have any gene sequence. And this was completely opposed to the philosophy of the Human Genome Project that has signed an agreement in 1996 to ensure that all information from the project would be made freely available to all within 24 hours after the release of the genome. So the conflict between public and private companies finally ended with a synergy between the two groups, and they announced together in year 2000 that they completed their working draft. And in 2001, these drafts of the human genome sequences were published simultaneously in 2 different articles. So the Celera approach, which is the whole genome shotgun sequencing, was quite similar to the hierarchical shotgun approach used by the international and public consortium. That public consortium employed, in fact, a 2-phase approach, first mapping and then sequencing the human genome. And this was required at that time because there was otherwise no kind of framework to work on in order to organize the sequencing. So the real difference between the 2 projects was that the public one used a map of the human genome, and this map served as scaffolds on which to assemble the DNA sequences. So in the first phase, that is called the shotgun, the chromosomes are divided into DNA segments that are then further subdivided into smaller DNA fragments that are overlapping, and then these are sequenced. These efforts were quite expensive, but were essential at that time for generating an accurate sequence. So the private effort bypassed the entire step of mapping, and instead the DNA was fragmented and modified by restriction sites and then cloned into plasmids, and then these plasmids were directly sequenced. So the quality of a sequence depends on the average number of times that each base of this genome is read during the sequencing process. So for the Human Genome Project, for example, we had a draft sequence that covered about 90% of the genome with a high accuracy, and a finished sequence covering more of the genome at a higher accuracy. So producing truly high quality finished sequences by this definition is very expensive. In fact, most human genome sequences that are produced today are draft sequences that are sometimes above and sometimes below the accuracy that is defined here. And this might explain why you can find several releases of a genome in the databases. And what I'm saying here is for the human genome, but also for any kind of genome that you can want to access, that you would want to access. So there are variable degrees of completion of published genomes. So there are the draft sequencing that processed in 2 distinct phases. So first is the high throughput and shotgun phase that we've been seeing. Then the data is assembled using one of the several available algorithms. And these algorithms will help to overlap the individual sequences on the basis of their similarity. And this results in the accumulation of about 80% to 100% of the original DNA sequence, typically in the form of contigs. Then the next step in the large-scale process is often referred to as finishing process. So in this step, you have the segments of sequences that are ordered and linked to one another, and you have a quality check, you have fewer, if any, gaps, and you get rid of ambiguities or discrepancies about segments, order, and orientation. So at this stage, any suspicious assembly is analyzed and either verified or double-checked or disassembled. And then, of course, you have the complete genome. So what you need to keep in mind is that obviously finished data, or having in hand finished data of the highest possible quality, is what you would ideally want to obtain from your sequencing. But a draft quality sequence can also be a good resource for many genomic experiments, like, for example, for genomic pathogens or unknown pathogens, for which even a draft would be a good amount of information. So what is important for you as end users is that you have a realistic understanding of data quality and the genomes you are dealing with. So why we are talking about the human project today? In fact, it's because the heritage of this project is huge. So there is a free access to various databases and resources for the scientific community. There is dedicated databases for various data and model organisms, and we've seen some of these like NCBI and Ensemble. And there is also community efforts now for non-model organisms like eukaryotic pathogens. You have an example of these databases here, and also a huge number of computer programs that are being released. Another really key finding of the human project was that the human genome contained very small number of genes compared to what was expected for that higher eukaryote. So consequently, there was a huge proportion of non-coding DNA, and researchers found out that the non-coding proportion was growing with the complexity of an organism. So basically, higher and more complex organisms contained an increased proportion of non-coding DNA. So detailing this would be beyond the scope of today's presentation, but this was just to let you know that there is a huge heritage of the Human Genome Project that has allowed the development of satellite projects, among which the ENCODE and GeneCode projects, in order to enhance our understanding of the human genome. So these are these projects, and you have the link for them if you want to know more about them. So the ENCODE aimed at generating a catalog of biologically informative representation of the human genome by using high-throughput methods and catalog all the functional and non-coding elements. And the GeneCode was to focus on the manual annotation of and characterize the gene features. You have the link to them if you want to know more about them. What I wanted to tell you also is that there was an international project that was developed also after the Human Genome Project. Because the goal of that project was to develop a haplotype map of the human genome. So basically it was identifying and cataloging most of millions of SNPs estimated to occur commonly in the human genome. So SNPs are single nucleotide polymorphisms. So these are genetic differences in individual bases of a genome that are by far the most common type of genetic variation. So the goal was to describe these variants, their location, their distribution among people from several different kind of populations. And it was designed to provide information to link genetic variants and the risk for specific diseases. And in fact, the 1000 genome projects nowadays has used many of the same population and extended the efforts of the HapMap project and has even replaced it. So the HapMap will be discontinued nowadays. And you have, so the 1000 project here, you have the link for it. So the goal was to identify most genetic variants with frequencies of at least 1% in the population studied. The project relied extensively on the development and sequencing technologies. The data that is contained in the 1000 Genome Project was of 2,500 individuals among 26 populations. And there is an international genome sample resource that was established to ensure the ongoing usability of data. So this is called the IGSR. And you also have other kind of resources like UK10K that aims at identifying rare genetic variants through the study of nearly 10,000 people, and in order to establish the link between genetic variants and rare diseases. So you have the link for them if you want to know more about them. So the development of novel technologies was also a heritage of the human genome, and they appeared as a consequence for it. So the next generation sequencing technologies are the most important of these technologies, and since their development they have gained increased attention with a considerable potential application in both diagnostic and public health microbiology, and it revolutionized the sequencing process. So we'll be seeing in the next part how we move